home yet. If our bodies are working so hard that we can't meet the oxygen demand, we never get to this stage. But if we're breathing enough, right, or we're bringing enough oxygen into our lungs, then what we'll do is we'll take those pyruvate molecules from glycolysis and move them inside the mitochondria, into the intermembrane space. And we're going to take one of those carbons and break it off, create a carbon dioxide molecule, and stick that 2-carbon molecule onto coenzyme A. Because we broke a bond, we liberated an electron, and we were able to make NADH. And then inside, did you have a question, Carly?
No.
Okay.
I'm just.
Okay. And then inside the mitochondria, all the way inside the inner membrane, what we're able to do is take coenzyme A, drop those 2 carbons off onto oxaloacetate, and then spin it and completely break down the sugar molecule, right? We make 2 more carbon dioxides per pyruvate. We make 3 more NADHs. We make another FADH2, and then an ATP. Okay. So you guys, what we've done so far is we've made all of the stuff necessary to make a whole bunch of ATP, and that's what's going to happen in the next stage. But let me show you really quickly the enzymatic reaction in which we make NADH. So what happens is there is an enzyme that has 2 active sites, and there is some substrate that's carrying an extra electron. In this case, it might be like pyruvate. So pyruvate would bind to the enzyme at this point, at this site right here, and then an electron would be transferred from pyruvate onto NAD. So this enzyme is binding to both the substrate and to NAD+. That electron is transferred over, and then NADH is created. I'll shut this so that it's not quite so distracting.
Oh my God.
You guys want to watch?
Yeah.
Don't grow up kids.
Don't grow up on the
Okay, while the parade is moving past, I'm going to make some more board space.
I have a
Do you
question.
want to take a picture of this? Because I have to start erasing stuff.
Yeah,
Can you turn the overhead off?
Yep.
So when it says step 1, what is the step 1?
This is step 1 right
That's
here.
the step 1? Okay. So on here, there should be in the...
That should be, yeah, in the intermembrane space.
I didn't even know they had that.
Okay.
You need to be focusing on biology. Dude, first trick or treat for a big first.
Okay, so the next stage of cellular respiration looks like this.
This is stage 3.
Yeah.
Okay. I need to focus. Stage 3.
It's exactly what it is. It's an electron transport chain.
So Dr. Turner,
Yes, ma'am.
so that first part of the Krebs cycle is step 1. The very other side, the ending product, is that step 2?
So the first stage in cellular respiration is glycolysis.
Okay.
The second stage is the Krebs cycle.
Okay.
The third stage is this thing right here, which is an electron transport chain.
Okay. Thank you.
Yeah, and then the last stage is going to be this stage right here.
Why is there one added?
Right, it's a good question. So there is one more cytochrome protein in cellular respiration than there is in photosynthesis. Functionally, it does the same thing. There's just more cytochrome protein in cellular respiration.
Wait, so what is the third?
I'm sorry?
What is it? So what is that then?
This is an electron transport chain.
I mean, like, are we going back to the photosystem or like what?
No, so this electron transport chain is embedded in this inner membrane
Oh, okay.
of the mitochondria.
There's always that one.
I'm sorry?
There's always that one kid that's
crying.
crying.
Unhappy.
Everyone else is having a blast, and there's that kid here. Oh. No, I'm just saying there's always like
Okay.
one kid that's crying for. I don't know why. But is it because
So, what happens, right, is an electron is traveling down the electron transport chain. And the purpose of an electron transport chain is what? To create a proton gradient. That's all we want to do. Where did we get that electron from in the light reactions? Yeah, so if it was the electron transport chain that was following photosystem 2, that electron came from P680, right? If it was following photosystem 1, then it came from P700. But we don't have those here, and so where are we going to get electrons? That's NADH and FADH2. So what happens is NADH comes over here, and it drops an electron off at the top of the electron transport chain. Was NADH just oxidized or reduced? It was just oxidized because it lost an electron. And that electron travels down the electron transport chain. And what we're going to do is we're going to take protons, right? And I should have showed this embedded in the membrane of the mitochondria. So this is located right here in this inner membrane. So that inner membrane is huge, right? And it's huge so that we can have lots of electron transport chain and lots of this stuff right here. Okay, so what we're going to do is we're going to take, did you have a question,
No.
Sydney? Okay. We're going to take protons from one side of the membrane, and we're going to pump them to the other side of the membrane. So that we're creating a strong proton gradient on this side of the membrane.
Where did those protons come?
That is a wonderful question. Those protons are coming from a lot of the oxidative reactions that are occurring right here. Where we're breaking up bonds and creating electrons.
Is this technically happening at the exact same time as that? If that makes sense?
Yeah.
Or is
Can-
it getting
it certainly
triggered
can
by
be.
the end of that?
It certainly can be.
Oh.
Yeah, for sure. So when we're pumping protons, what we're doing is we're pumping protons from inside here to the intermembrane space. And we're creating a very strong concentration of protons in between mitochondrial membranes. Okay, so the next stage, this stage of cellular respiration, is the electron transport chain. And the purpose of the electron transport chain is, to create a proton gradient. Is that an active or a passive process? It's close to passive. Yeah, it's active. It's active because we're using electron energy, right? And what we're doing is we're taking something from a low concentration and pumping it to a high concentration. So it has to be active. It has to require some energy. So the products of the electron transport chain is what? What did we make?
An AD plus.
We made some NAD pluses, yep. But what's the actual important product? It's the proton gradient. That's all it is. The product is the proton gradient. Did we make any ATP? None. All we're doing at this point is making a proton gradient. But why do we want that proton gradient? In photosynthesis, following photosystem 2 and its electron transport chain, what did we use that proton gradient for? To create a bunch of ATP, right? So that's what this proton gradient is going to be used for here. We're going to use that proton gradient to turn ATP synthase and make a whole bunch of ATP. So this is the last stage in cellular respiration, and we're going to call it chemiosmosis. Okay, now the way this happens, right, is we take these protons and they just fall back through ATP synthase. Like that. And we know that every time a proton falls through ATP synthase, it spins. And that kinetic motion, the kinetic energy, is harvested and used to create ATP. So we get ADP. Plus an inorganic phosphate to make ATP. Now, per glucose molecule, there is a range of ATPs that can be created. We'll talk about why there's a range in just a few minutes. But generally speaking, you get approximately 34 to 36 ATPs created at chemiosmosis, so a ton.
What happened to that electron that fell through?
Good question. That electron, plus some of the protons and some of the oxygen that we're breathing, becomes metabolic water. And that's where we're making our 6 H2Os.
From the equation.
From the equation. Exactly right.
Makes sense.
Isn't it great when that happens?
Yeah.
It's all too infrequent, but it's nice when it happens. Okay, so the purpose of chemiosmosis is to create lots of ATP. And the actual products, the ones that we care about at this stage, are ATP, and there are 34 to 36 of those, and then also some water, and there are 6 of those. You know, what's crazy is there's some animals that live in deserts. Have you guys ever heard of kangaroo rats? They're super cute. It doesn't sound like it because they're a rat, but they're pretty cute organisms. And they don't drink any water. They're able to get all of the water that they need by metabolizing the seeds that they're eating. So they're able to generate enough metabolic water at this stage to meet all of the demands of their physiology. Okay. So, you guys, when we're creating ATP at chemiosmosis, is that oxidative or substrate level phosphorylation? It's oxidative. And so, in which stages, glycolysis, Krebs cycle, electron transport chain, chemiosmosis, in which stages are we creating ATP by oxidative phosphorylation? Just this one. This is the only place where we're creating ATP via oxidative phosphorylation. All the rest of the ATP creation is substrate level.
Can you repeat that?
You betcha. The only stage in cellular respiration in which we create ATP by oxidative phosphorylation is chemiosmosis.
And is that because you bring the inorganic phosphate?
That's exactly why. Because these phosphates that we're using here are all inorganic. They're all dissolved in the inner sanctum of the mitochondrion. Yeah, Adam.
The only stage we use oxidative phosphorylation is like
To create ATP.
to create ATP is
Osmosis.
osmosis.
Exactly right.
Are these last two stages, they're in the inner membrane?
Yep, all of these proteins are embedded in this inner membrane. So, I have a question for you. We've talked about photosynthesis and we've talked about cellular respiration now, and this is the entirety of these processes. We know that we go through cellular respiration. Do plants go through cellular respiration?
Yeah.
Yeah. For sure. I mean, they photosynthesize for a reason, right? I mean, they photosynthesize to create sugars so that they can create ATP. But why does a plant need ATP? I mean, it's not jogging a mile.
Is there living things?
And living things do what?
They create
They
oxygen.
grow and they reproduce and they repair themselves, and all of those metabolic functions require cellular energy. But think about it. What plants do, right, is they go through the light reaction.